Hello, my name is Caitlin Schudark, and today we're going to talk about how depolarization occurs throughout the heart and the mechanisms behind cardiac muscle contraction. This is the heart. We have the atria and the ventricles. Electrical signals propagate through the heart. This is a phenomena referred to as depolarization. Depolarization is a cascading effect of the cells in the heart becoming more positive. So from negative to positive. Within the cardiac myocyte, there are intercalated discs. These are conductive bands that allow the signal of depolarization to be propagated throughout the heart. This process starts at the SA node, or the pacemaker of the heart. The SA node begins the process of depolarization because it has automaticity, meaning it can depolarize on its own. This is going to cascade a signal throughout the atria, eventually reaching the AV node. After the AV node depolarizes, it is going to travel down the bundle of His, break off into the right and left bundle branches, and then wrap back up around the heart through the Purkinje fibers. This wrapping of the signal allows the ejection of blood to go up and out of the ventricles. So to review, so far we know that an electrical signal, also known as depolarization, or the directionality of positive ion flow, starts in the SA node, travels through the atria to the AV node, down through the bundle of His, to the right and the left bundle branches, and then back up around the heart through the Purkinje fibers. Now we must look at how this depolarization actually causes a contraction. To do this, we must zoom into the cardiac myocyte. This right here is part of the cardiac myocyte, with this being the T-tubule. So in the T-tubule, our depolarization is going to cause a small release of calcium. This small trigger calcium is going to travel here through the ryanodine receptor, through the feet, and it's going to cause a much larger calcium release from the sarcoplasmic reticulum inside of the sarcolemma. This release of calcium from the sarcoplasmic reticulum is going to come and bind with troponin C. This binding of calcium to troponin C is going to move this tropomyosin off of the actin, which is going to show the myosin binding site on the actin molecule. So it's going to twist away that tropomyosin, revealing the myosin binding site. Actin and myosin are the two filaments associated with the actual shortening of the muscle fiber that occurs during a contraction. This is accomplished by one of the two myosin heads ratcheting and binding to the actin, pulling the actin and shortening the sarcomere. Another way of viewing this is in this diagram here. We can see our actin, we can see our myosin, and we have our titin that's anchoring it in place. Now this myosin is going to ratchet up our two heads, and it's going to pull along, shortening the overall sarcomere. This occurs once calcium is added, binding to that troponin C. We can see the full length of the sarcomere shortening, which is what's going to actually cause that shortening of the muscle for the contraction. This shortening of the muscle fibers are going to continue until calcium is removed. This is going to bring this shortened sarcomere back to its original length. Calcium is removed by SERCA, this protein right there. SERCA is actually going to pump calcium back in to the sarcoplasmic reticulum. Once the calcium is pumped back in, there will be no more calcium binding to that troponin C, allowing the tropomyosin to move back over the myosin binding site on this actin. This will allow the cardiac muscle to relax. This contraction that's triggered by depolarization is how force is exerted, and how the SA node is able to trigger a synchronized contraction across the atria and down and back up around the ventricles, allowing the heart to sufficiently pump blood. Now that we know how depolarization and contraction occurs in the heart, what would happen if we inhibited SERCA with a drug? Now thinking back to the calcium pump SERCA, we know that SERCA is responsible for pumping calcium out of the cell, the sarcolemma, and back into the sarcoplasmic reticulum. If there was a drug that inhibited SERCA, meaning it was no longer pumping that calcium back into the sarcoplasmic reticulum, that calcium would be stuck out here in the sarcolemma, continuing to bind to this troponin C. If calcium was keeping to bind to troponin C, tropomyosin would remain away from the actin-myosin binding site, meaning the sarcomere would continue to be shortened, and this would result in both a stronger contraction and likely the inability of the cardiac muscle to relax.